Hi, good evening. Welcome to Biochemistry Class 6. Welcome back to our summer sessions. Today we're going to be going over Biochemistry Class 6, which is about biologically important molecules. This is our second installment of biologically important molecules. And this time we'll be going over carbs and lipids, carbs also known as sugars. And my name is Karen, and I hope you enjoy this presentation. So, beating the dead horse, we're Pre-Med Peers. We're a nonprofit in the Commonwealth of Virginia. We're here to help you always, and you can always contact us through Slack or email. This session is currently being recorded, and the slides will be provided, so you can always refer back to these if you didn't get anything down in real time. Here's our info at premedpeers.org. That's our email, and yeah. I think we have a new person. Well, Jennifer, if you ever come in, welcome. Here's our session outline. So we're going to first be going over the homework. It should have been from Khan Academy and mcatreview.org pertaining to carbohydrates as well as fat metabolism, carbohydrates and fat. And then we'll be doing content review. And then we're going to go over some warm-up questions, hopefully to jog your brain and to test what you just learned or reviewed. And then we're going to do a passage on glycogen. Hi, Jennifer. What's up? I'm about to start the homework review. If you had any questions on that. And this is our content outline. We're going to be going over these topics under sugars or carbs, and we'll be going over these topics really quickly under lipids. The sugars one is a lot longer. And so now it's time for homework review. If you have any questions, feel free to put it in the chat or shout them out. I did the questions for Khan Academy on the carb side, and I have screenshots of them. So if you have any questions about those, I can always pull it up and we can go over it together. So Jennifer, did you have any questions on the Khan Academy stuff or the MCAT review stuff? If not, I'll just move on. Jennifer, feel free to interrupt me at any time. And so now we're moving on to content review. We're going to be starting off with carbohydrates/sugars. So here's the basic structure of a carbohydrate or sugar. Generally, there's 3 requirements that you need to meet in order to be classified as a sugar. You need to have a minimum of 3 carbons in your carbon backbone. You need to have a presence of either an aldehyde or a ketone group. So if you can see in this little family, all of these sugars have aldehydes on top of them. So on carbon 1, there's an aldehyde group. So that goes for every single sugar in this picture. In addition, you also need to have at least 2 hydroxyl groups present in your structure. So you can see here, glyceraldehyde meets the bare minimum. It has 3 carbons. It's a 3-carbon sugar with 2 hydroxyls and an aldehyde group. And so that's the bare minimum. And then you have your 4 carbon sugars here on the second row, your 5 carbon sugars on the third row, and your 6 carbon sugars on the fourth row. I'll be pointing out which ones are the most important for you to know. So don't worry about memorizing all of these. Next, we'll be going over this kind of comparing the 2 different kinds of structures that you'll usually see with sugars. We have the Fischer structure on the left side here, which is a linear structure. And we also have the cyclic structure, also known as the Hayward structure. So I have here in this picture, these are both glucose, but they only look different because they're structured different. So you have your Fischer on one side and the Haworth on the other, but they're both glucose. And so when it comes to like figuring out like the structure, knowing how to number the carbons and knowing how to figure out which carbon is which is extremely important. So when you're looking at a Fischer structure, you would count the carbons from the top to the bottom. So starting with this aldehyde up here, carbon 1, then carbon 2, 3, 4, 5, and 6. Carbons 2 through 5 are considered chiral carbons. In the Hayworth, most generally, you'll see it in this kind of orientation, upright with the carbon 6 on top. And in this case, you would count in the clockwise direction. So here's carbon 1, also known as the anomeric carbon. Carbon 2, 3, 4, 5, and 6 going clockwise around the sugar. Also, some other things you need to know in order to kind of translate between Fischer and Hayworth structures. Generally, if you have an OH group on the right side of a chiral carbon, it'll translate to a downwards position on the Hayworth. So as you can see here, you have 2. 2 is on the right. 2 has a hydroxyl group on the right side. And so now on the Hayworth, it's pointing down. So moving on to carbon 3, it's pointing to the left. And generally on the Hayworth, that translates to being upright. So here you have the carbon 3 with the OH group on top. And so there's 2 ways I like to remember it. There's like a phrase I like to say in my head, like, for example, like these sugar structures are downright confusing. So you can remember, oh, down goes with right. And Hayworth is almost always down and up, and Fischer is pretty much always right and left. Another thing I do is I take the Fischer structure and I rotate it 90 degrees clockwise in my head. And then from there, you can see, oh, okay, the 2's down, the 3's up, the 4's down, the 5's down. And it translates pretty smoothly to the Hayworth structure, even though it's not shaped like a hexagon. So yeah, so that's structure. And I also just wanted to point out, you might once in a while see a chair conformation of sugars. I just wanted to point it out. You'll mainly see the first 2. But one thing I wanted to note is that so in the chair conformation, you have the axial and equatorial positions. And the up and down of Hayworth doesn't necessarily translate directly to axial and equatorial. It really depends on which carbon you're looking at. So for example, for carbon 3 in glucose here, you have the OH pointing up, and then the OH is pointing up here, but it's equatorial. And you have the carbon 4 is pointing down in the Hayworth, and it's pointing down technically in the chair conformation, but it's also equatorial. So you have to be really careful to see which way the line is actually pointing, and not trying to make a strict association between down and up versus axial and equatorial, if that makes sense. Next, we're going to go over some stereochemistry. So there's generally 2 concepts that you really need to know. So there's the absolute configuration of D and L. And generally D sugars, you're almost always going to see D sugars. They have the hydroxyl group of the chiral center farthest from the carbonyl group is always on the right side. So D is right side, and then L sugars, which you'll rarely see, is on the left side. But that's easy to remember, right? So it's like, oh, L, that means the hydroxyl group of carbon 5 is on the left side. And then you have your kind of anomeric stereochemistry. You only see this alpha and beta distinction in cyclic structures only. So in Hayworth or in chair, like you see here, the alpha anomer, you can figure out that it's an alpha anomer by comparing carbon 1 generally to carbon 6 and studying their positions. So if, for example, if carbon 1 has their hydroxyl group pointing down, and the carbon 6 OH group is pointing up, that means they're trans to each other, so they're pointing in opposite directions, and so that makes it an alpha anomer. On the other hand, your beta anomers, they have their hydroxyl groups on C1 and C6 generally, pointing in the same direction, so they're cis. There's also 5-ring structures, so in that case, it would be carbon 5 instead of carbon 6, but you're mainly going to deal with carbon 6 structures. Next, I also wanted to go over just to explain how exactly sugars go from Fischer to Hayworth. And using organic chemistry speak, there's a nucleophilic attack going on between the OH group of the second to last carbon, so in this case, either C5 or C4. So the hydroxyl group here is the nucleophilic, is the nucleophile, and it goes and attacks it reaches over and it attacks the carbonyl carbon on carbon 1. So as you can imagine, because it's a carbonyl, the oxygen is pulling electron density away from the carbon, making it more electrophilic. So that's why it would make the most sense, why the carbon would be most likely to be the electrophile. And the reason why the OH group has to be on the second to last carbon is so that it can form a sterically stable ring. Because if you take it from any other chiral carbon, it won't make a very stable ring. The most stable rings tend to have 5 members or 6 members. 4-membered rings, which are like square-like, they're not very stable. 3-membered rings, which are, it looks like I'm getting a call. I'm just gonna hang up. That was weird. I got like a call from somebody on Slack. Anyways, so 3-membered rings, they're like in a triangle shape, not very stable either. So during this nucleophilic attack, so you have the hydroxyl group making a bond with the carbonyl carbon on carbon 1. You can see it more clearly on this 90-degree rotated diagram on this side, on the right side. Oh, give me 1 second. Sorry about that. So you have the formation. So you can see in the Hayworth projection here, you can see more clearly the connection between the first carbon, the anomeric carbon, and carbon 5's OH group. So carbon 5's original OH group. And then up here you have carbon 6, so that's CH2OH. So that's carbon 6's hydroxyl group. So you can see it more clearly here. So we're letting a new person in. Hi, Rashina. I'm going over sugars right now.
Hi, oh my God, I was like on the wrong link, I think, for like 30 minutes.
Oh no, I'm so sorry. It was probably like it might have been the July one, huh? Yeah, I'm sorry. The code
No,
always
I
changes
was on the
every month.
link that they sent, that you guys sent today. Like this, I guess the September link. They sent out a new link today.
Oh, yeah, they sent it today. Yeah.
Okay.
Yeah, sorry for the inconvenience.
That's okay, don't worry about it.
Yeah. So moving on in the presentation. So if you have a 6-membered, if you have a six-membered Fischer structure, it forms a pyranose or a six-membered ring. Or if you're dealing with a 5-carbon structure, you'll end up forming something called a furanose or a 5-membered ring. And these terms are important to note for classification. And speaking of classification, every single sugar and carb you'll see, their scientific name almost always ends in ose. So you have aldose, ketose, triose, pentose, and hexose. Well, what exactly do they all mean? So there's usually 2 formulas that you need to pay attention to. Here's 2 that I'm pointing out. Sometimes you'll see a sugar named for the number of carbons they have in their backbone. So in this case, triose has 3 carbons in their backbone, so tri3. You'll also see sugars being named with the number of carbons, as well as whether they contain an aldehyde or a ketone group on their first or second carbon. So in this case, aldotriose. So a glyceraldehyde is an aldotriose because it has 3 carbons and it has an aldehyde group, and it contains an aldehyde group. So aldose, aldehyde group, and ketose, ketone group. Pentose means 5, so 5-carbon sugar. Hex means 6, so 6-carbon sugar. And yeah, and then just in case you forgot, chemical formula of sugars is always CnH2nOn. So there's always twice as many hydrogens as there would be carbons or oxygens. Next, I'm going to go over some common sugars that you're going to be seeing. I'm going to start with monosaccharides. I'm going to be going over 3-carbon structures, 5-carbon structures, and 6-carbon structures. So let's start with glyceraldehyde. So glyceraldehyde is an aldotriose. It has an aldehyde group on carbon 1, and it has the OHs pointing on the right here. So for all the ones with the OH on the right over here, just assume that they're all D format, so they're a D configuration. So there's glyceraldehyde, and then there's dihydroxyacetone, which is a ketotriose, and its ketone is located on carbon 2. Most ketoses have their ketone on carbon 2, so keep that in mind as well. So usually the aldehyde is on the first carbon or the ketone is on the second carbon. I always get kind of thrown off visually, but it's important to have in mind. And I'm going over these 2 because, you know, they sound really familiar. Almost forgot where it's from. I'm just kidding. It's from like the pentose phosphate pathway and stuff like that. Glycolysis, the carbohydrate metabolism stuff that we went over in the very beginning of our biochem series, because that's some of the most important stuff to know. Next, we have our 5-carbon structures, ribose and deoxyribose. They should also sound very familiar to you. Ribose has 5 carbons. It has an aldehyde, so it's an aldopentose, and all of it in the D configuration, all of its hydroxyl groups are on the right side. And so you can see here in Haworth projection, you have your OHs pointing down here, and then the oxygen here becomes this oxygen on the top, and then carbon 5 has its own group here. And it's alpha because the hydroxyl group here on the anomeric carbon and the carbon 5 hydroxyl group are trans to each other, so they're in opposite sides. So that's alpha, and then just going it over again, and then beta is on the same side, so they're cis. So that's beta D ribose and then alpha D ribose. You'll always put the alpha or beta before the D or the L. So that's kind of how you phrase it if you're trying to like write it out or something. On the other hand, you have 2-deoxy D ribose. The only difference between the 2 is that there is just a hydrogen in place of the hydroxyl group, which should sound very familiar to you. But this is what it looks like in Fischer projection. I I think when we learn about nucleotides, we see a lot of it in Haworth projection just because of that backbone. But yeah, it's interesting to note the Fisher version of it. And then here is like your holy grail monosaccharides, the 6 carbon structures. You have glucose, fructose, galactose, and mannose. Those are the ones that were the most high yield on the exam. And starting from what kind of structure they are. So glucose and galactose and mannose, they're all aldoses. They're all aldohexoses. On the other hand, fructose has a ketone on carbon 2, so that makes it an ketohexose. And the way I like to, these look really familiar, and I remember this is one of the things I hated learning because it's just rote memorization. You just have to know what they look like. And so I wrote here some notes on like how I remembered what they looked like in a way that's independent of whether it's a D or L or if it's an alpha or beta configuration. So with glucose, I want to point out that you have carbon 2 and carbon 4, their hydroxyl groups are on the same side. And then galactose is an epimer of glucose. You could also, another way you can approach it is to memorize glucose and then to memorize what's different about everything, what difference, what the difference everything else has with glucose. So comparing everything to glucose. So you have galactose is an epimer for carbon on carbon 4. So in this case, you have carbon 3 and carbon 4, their hydroxyl groups are on the same side. For mannose, mannose is an epimer of glucose at carbon 2. So that means the hydroxyl groups for carbon 2 and 3 are on the same side when it comes to mannose. And I'll explain what an epimer is in the next slide if you don't know what it is already. And you have fructose here, it's on carbon 2. But for the most part, so you have your CH2OH on carbon 1 and then your C double bond O on carbon 2 because it's a ketose. But the rest of the structure is identical to glucose. So if you're having trouble remembering the rest of fructose, just kind of think of glucose. So yeah, so that's kind of, those are the kind of strategies I would suggest for memorizing these things. And also just like make flashcards. Flashcards are really helpful. Getting things wrong always helps you, even though it hurts. And it's also, I didn't have any time to put the Haworth projections of all of these up, but learn to know what these look like in Haworth because it'll pay off and you'll see in the next couple slides. So going over, so an epimer, what was I talking about when it came to an epimer? So before I talk about epimers, I want to talk about enantiomers and diastereomers, like that classic organic chemistry concept, because epimers and anomers build off of this concept. So enantiomers, if you already know, they differ at every chiral carbon between the two sugars. So I picked a really simple example, glyceraldehyde. It only has 1 chiral carbon, but you know, it's on the opposite sides. So the enantiomers, they form a mirror image to each other. So of course here you have the OH on the right side for D-glyceraldehyde, and then OH on the left side for L-glyceraldehyde. So with that in mind, D and L structures for the same sugar, they're always going to be enantiomers. Diastereomers, this is like a family of things that differ at least 1 chiral carbon. So you have your ribose, your classic 5-carbon structure, and you can see here arabinose, it differs at 1 carbon. Xylose, it differs at this carbon, and then lexose, it differs at 2 carbons. So that means they're all diastereomers. Epimers, they differ at only 1 chiral carbon. So bringing it down, so going further, you would identify arabinose and xylose as epimers specifically, and not just diastereomers. you see here with glucose and galactose, which I already pointed out as well. And anomers, they're a specific kind of epimer because they defer at only the anomeric carbon. So that's the carbon you should be looking at when you hear anomer. You look at the anomeric carbon and see which direction the hydroxyl group is pointing. And so another thing to know is that all alpha and beta configurations of the same sugar, they're anomers. So, but they also have to be the same. They also have to be both D and both L. So they can't differ. Yeah. So it has to be consistent on that end at least. But the alpha and beta of the same sugar and the same D or L configuration, pretty much always anomers. Moving on from monosaccharides, we got our disaccharides, and they're always going to be shown. I have never seen them in Fisher projection. They're always going to be in Hayworth. And disaccharides are made up of two monosaccharides. So you kind of see why you need to memorize the monosaccharides in Hayworth, like because the disaccharides are in Hayworth. So you have to, they're going to throw disaccharides at you. I'm sorry. Like you're going to have to know what they look like. So the top three disaccharides you need to know are sucrose, which is made up of glucose on the left side and fructose on the right side. You can tell fructose is fructose because of the difference because you have the CH2OH on this side because it's a ketose. And then lactose is made up of galactose and glucose. And maltose is made up of glucose and glucose. So if you ever see a disaccharide and you see that the monosaccharides look pretty much identical, they're probably both glucose, and you're probably looking at maltose. So they named this image in particular, they named it with kind of what's on the left side and what's on the right side. But it doesn't always, you're not always going to see it that way. Sometimes you're going to see long chains of sucrose. You're going to see long kind of polysaccharides containing like sucrose or lactose and maltose. And you're going to have to be able to identify which monosaccharide is which. So that's important. Disaccharides, they contain something called glycosidic linkages. So kind of this hand-holding looking thing in the middle between these two monosaccharides, that is a glycosidic linkage. It's a linkage between two glycosides or sugars. There's two types of linkages you'll be seeing. There's an alpha linkage that kind of looks like the classic like hand-holding. And they're named for pretty much their shape. So the alpha with the purple maltose has kind of, they're both kind of going down like this. And the beta glycosidic bond, you have one, it's a connection between two OH groups that where one of them is pointing down and one of them is pointing up. And so you kind of have like this snake looking bond that happens instead. So yeah, it does depend on the configurations on which way the OH is pointing for the respective monosaccharides. There's also the two most common carbon linkages that you'll see are between either carbon 1 and 4. So carbon 1 is the anomeric carbon. So carbon, let's see what this one is. So let's look at maltose on this left side. So you have your glucose on the left side. Okay, it has a carbon 1. So there's a carbon 1 on this side for the linkage. And then counting on the right side, you have 1, 2, 3, 4. So this is an alpha 1,4 glycosidic bond because it's between a carbon 1 with one sugar and a carbon 4 on the other sugar. So that's that. And then you're also going to see glycosidic bonds between alpha 1 and alpha 6. I'll point that out when we get to polysaccharides, but that's also between, so it's between the anomeric carbon and carbon 6 of the other carbon, of the other sugar. Really quickly, I want to go over something called Benedict's test. Benedict's test identifies something called a reducing sugar. So like, what's a reducing sugar? So let's look at these disaccharides here with comparing lactose, maltose, and sucrose. Reducing sugars, they tend to have something called hemiacetals, which you should have learned already from organic chemistry. They have something called hemiacetals. So why is that important? Hemiacetals, they're able to make a bond to another sugar. And it's also easy for them to break their cyclic form. And non-reducing sugars, they don't have hemiacetals, they only have acetals. So if you can see here in these bottom photos, you have the hemiacetal and the acetal. On maltose and lactose, they have both, but because sucrose only has acetals, it's locked in its cyclic form, if that makes any sense. So Benedict's test, it identifies reducing sugars by detecting free aldehyde or ketone functional groups that become present when the sugar is reduced. And Benedict's test is color-based. So you have on the top here, you kind of have the gradient between, like what color should I be looking for if we're testing a non-reducing sugar, and like what color should I be looking for when we're testing reducing sugars. So usually when reducing sugars are present in the Benedict's test, you'll see the formation of a reddish precipitate. And in a negative Benedict's test, which means that there's no reducing sugars present, you'll see the color won't change because the reaction that occurs during Benedict's test when it detects when it detects a reducing sugar, the chemical reaction that occurs causes it to turn red. I won't go into too much detail, but yeah. But if there's no reducing sugars present, it doesn't change color at all, so it stays the original color that it has. It could be blue, it could be a different color, but it's usually blue. And so, but the main point I have with this is kind of to remember which sugars are reducing and which are non-reducing. Lactose and maltose, they're both reducing, and sucrose is a non-reducing sugar, a non-reducing disaccharide, which is very important to know. They'll probably test you on that. And then next we have polysaccharides. So we have our lovely starch and glycogen and cellulose. These are the three that you'll see the most. You can kind of see how they differ in their branching styles. So their branching styles differ based on whether they use alpha-1,4 glycosidic linkages or alpha-1,6 glycosidic linkages, or it could be beta as well. Or if it uses both. So on this chart here, you can also know where these polysaccharides are present. So starch and cellulose, they're plant exclusive, and glycogen can be found in animals like us. So we store sugars as glycogen if we really need to pack it up. And the bonds that they use differ. So cellulose, it only uses alpha-1,4 bonds, and you can tell because the lines for it are always straight. It's not branched because it can only branch in the horizontal direction in that linear fashion. As opposed to starch and glycogen, they have the ability to use 1,4 and 1,6 bonds. So the the 1,4 bonds, they go this way, and the 1,6 bonds, they go kind of vertically. So that's what lets them branch out. So I think it's amazing. So you have kind of more like diagrams in this chart, and then kind of the impact of the branching on kind of the overall shape of this thing. Like, look how pretty these are. Another thing I want to go over really quickly is the enzymes that are related to it, because you might be tested on the kind of the buildup and breakdown of these polysaccharides. So starch is broken down using amylase, cellulose is broken down using cellulase, and glycogen has two enzymes to build it up and two enzymes to build it down. So glycogen synthase allows for the connection of 1,4 bonds, and glycogen branching enzyme, because to be more vertical, glycogen branching enzyme focuses on 1,6 bonds, so it builds 1,6 bonds. Glycogen phosphorylase breaks down 1,4 bonds, and glycogen debranching enzyme breaks down 1,6 bonds. So you can kind of see how these pair together in a way. So synthase and phosphorylase, and then branching enzyme and debranching enzyme. That second pair is pretty obvious. The last thing I wanted to go over with carbs is something called mutarotation. Mutarotation is, you might see this somewhere. It is a change in optical rotation due to a change in equilibrium between two anomers. And it happens when there's two different anomers and their chiral centers start to like convert from like one to the other. And so they're like becoming each other, and they're like kind of like switching places in a way. So you have this example here with kind of like alpha D-glucose and beta D-glucose. And they're both being dissolved in water. And they have different specific rotations originally. But over time, you'll see their specific rotation approach the same number. And that's because of that interconversion in chiral centers. So it's mutarotation. Muta means change. So means change of rotation. Yeah, and that's all I had for carbs. Did anyone have any questions before I move on to lipids?
No.
Okay. Regina, do you kind of get everything what I'm saying?
Yeah, everything was good. Yep, perfect.
All right, sounds great. So lipids, this one is a shorter section than carbs. So I'm just going to go over like the really basic structure of lipids that you're going to see on the MCAT. So fatty acids are consistent of one carboxyl group and one hydrocarbon chain. So I really like this graphic because it shows it very clearly. That's the general structure of a fatty acid, and that's literally it. So the carboxyl group is the polar side, and the hydrocarbon chain is the nonpolar side. And then you also have the distinctions between, saturated fatty acids, unsaturated fatty acids, kind of like the difference in like the cis bonds and the trans bonds and unsaturated fatty acids. You have your polyunsaturated fatty acids and your monounsaturated fatty acids, but that's just like the basic difference. Saturated fatty acids have all single bonds, and unsaturated fatty acids, they have their kind of like double bond somewhere. So that means they're not fully saturated in hydrogens. That's kind of how I remember it. So yeah, that's the difference that we should already know already. And triacylglycerols, or tags, they build on fatty acids. So tags have a glycerol backbone, which is polar, and it has 3 ester bonds to 3 fatty acids. And being able to store 3 fatty acids, when you put all of that together, that makes for a very energy-rich storage mode. So triacylglycerols, their function is to store energy. And this also makes them very hydrophobic. So despite their glycerol backbone, you have all these hydrocarbon chains here. So it's hydrophobic. Next, I kind of wanted to go through this chart of kind of other lipids you might see. These are kind of like your backbone lipids. So like phospholipids, sphingolipids, glycolipids. These are the things that you'll see in your cell membrane. So you got that classic like head with the 2 tails. That's your phospholipid, more specifically, most likely your glycerophospholipid. So everything pink here, so you have your triacylglycerols to compare. Everything pink in this diagram is the backbone. So you see here glycerol backbone, glycerol backbone. So phospholipids are lipids that contain a phosphate group as part of their tail. And phospholipids can be split generally between glycerophospholipids and sphingolipids. And it's broken down between the difference in the backbone. So you have your glycerophospholipids, it has a glycerol backbone, 2 fatty acids, a phosphate group, and an alcohol. A sphingolipid has the sphingosine backbone. So kind of remember this structure for sphingosines. I didn't have time to put the exact structure, but they're almost always shaped like an L. So pay attention to that. So in case you confuse it for excuse me, for a fatty acid chain. So make sure to pay attention to that. Sphingolipids, they only have 1 fatty acid, but they still have a phosphate. And choline here is an example of a head group. There's all different types of head groups. As you can see, I studied Kaplan for the MCAT, and I literally wrote all of this down. Like, they had like these different structures that I don't have time to go over right now. But this general schematic is the most important to know. So that's your phospholipids. And then you also have glycolipids, which mean they have a sugar in their as part of their kind of like tail collection. So under glycolipids, you have sphingolipids. So you have other sphingolipids with the sphingosine backbone and a fatty acid, but instead of a phosphate, you have a sugar. So yeah, so that's the difference between all 3. And then more lipids, we got our steroids. We got that classic 4-ring structure, 3 cyclohexanes and 1 cyclopentane. Steroids are found in the cell membrane, but not as much as the phospholipids and the sphingolipids and the glycolipids, which are like the literal backbone of it. That's like what you see when you see the circle. The steroids are kind of like dotted and are kind of like sprinkled within like the phospholipids. But they do influence the membrane's fluidity, kind of, and it influences the fluidity differently depending on whether the temperature is high or the temperature is low. It really depends. And they also act as hormones in the body, so they're signaling molecules. And then waxes, on the other hand, they're usually found in a lot of plants, and they're technically esters that are comprised of an alcohol chain and a fatty acid chain. So the alcohol chain would be on the right side here, and the fatty acid chain would be on the left side here. So you can see how it makes an ester through that middle structure. And waxes, they can be used to prevent water loss in plants. That's why they're so smooth and beautiful. And then real quickly, I wanted to go over how steroids are formed. I have cholesterol as an example steroid here. So it's a building block process. So you have your isoprene, which consists of 5 carbons and 8 hydrogens. And it has 2 double bonds here. The double bonds are the key to kind of building these branches of lipids, and it's also the key to kind of making sure that everything forms like the rings. So from isoprene, you put 2 isoprenes together and you make something called a terpene. And then there's different types of terpenes, but 3 terpenes, you can put 3 terpenes together to make a squalene. So that would be 6 isoprenes. And then from there, squalene they go through some other steps, and it like cyclizes to form cholesterol. So that's the general schematic I want you to know: isoprene, terpene, squalene, and then cholesterol. And then we have our signaling molecules. So I just wanted to mention, to just throw out some lipid signaling molecule names. There's a family of lipid signaling molecules called eicosanoids. I don't know if that's the way to pronounce it, but that's how I've always pronounced it. And they include prostaglandins. It includes things called prostacyclins, thromboxanes, and leukotrienes. I usually see prostaglandins the most. So kind of pay attention to what prostaglandin looks like. And they kind of look like squids. I don't know why they look like squids to me, but they do, because they have like the head here and like the legs. I don't know. And yeah, they're derived from phospholipids. So that might be pretty easy to see if you can like kind of imagine the backbone that used to be there. So yeah, that's icosanoids. And then on the right side, I just wanted to throw in some other steroid molecules. I taught the endocrine system earlier in July. And so these should look really familiar to you, or at least the names. But here's the structures in case you want to get familiar with the steroid structure. And that's it for lipids. That's all I have for lipids. So now we have our warm-up questions. Did anyone have any lipid questions before I moved on?
No. Oh, no.
Okay, sounds great. So I have 4 warm-up questions for you guys today. So question 1, which of the following accurately describes glucose? Feel free to put it in the chat or just say it out loud if you have an answer.
I want to say B.
Okay. Do you have any reasoning behind that?
I know it's a hexose. I'm just
Mhm.
I don't know if it's a ketone or aldehyde. I'm just gonna go with B.
Okay. Yeah, great guess. So it is an aldohexose. So glucose, I wish I had a picture, but you have tons of pictures in the PowerPoint. So aldo means that it has an aldehyde group. So glucose has its aldehyde group on carbon 1. And yes, it is also a 6-carbon sugar. So great job. Question 2 is about Benedict's test. A solution of an unknown sugar is subjected to Benedict's test, which leaves the color an unchanged blue. Assuming the solution contains a disaccharide, what are the 2 monosaccharides that compose it?
D.
D? Correct. Yeah. So because it stayed blue, it must be a non-reducing sugar, and sucrose is the only non-reducing sugar that we discussed. So yeah, and so sucrose is made up of glucose and fructose. Do you know what the other 2 disaccharides are for A and B by any chance?
Is it maltose for A? And lactose.
Yeah, great job. All right, question 3. Which is the following is false regarding steroids?
Is it B?
Yes, it is. So yeah, that's kind of why I discussed it like in the way that I discussed it before, is because I had it as a warm-up question. So so cholesterol, for example, a type is found in the plasma membrane, but it's not the lipid that comprises that's like the main backbone of it. Instead, it's composed of like phospholipids and like sphingolipids and stuff like that. Just to go over the wrong answers, yeah, steroids and triglycerides are both fats. Steroid hormones are lipids. So lipids have the ability to cross the phospholipid bilayer and be able to get to the nucleus and bind to receptors there, and kind of and have like really pretty direct signaling that way. And then all steroids contain at least 4 rings, the 3 cyclohexanes and 1 cyclopentane. And our last question, which of the following lipids is responsible for the storing of energy in the body?
A.
The answer is actually C. So glycogen is actually a carbohydrate, and it's sorry, I had like a brain fart. But glycogen isn't a lipid, so that would be kind of automatically wrong. Triacylglycerols are the one that stores energy in the body because it contains 3 fatty acids, and that's a lot of energy being stored in one molecule. So it has the largest potential to store energy. Glycerophospholipids, they're more found in the membrane, and then eicosanoids, which I went over before as well, is part of the signaling molecule family. So that's their main purpose. Yeah. So
Can you
that's it
just
for
leave
the
this for
warm-up
1
question.
second? Oh, can you go back 1 second?
Yeah.
So triacylglycerols, acids, and has the largest. Oh, so triacylglycerols are lipids, and they
Mhm.
store energy. Okay.
Yeah. Yeah. So the only one that wasn't a lipid was A, but the rest are all lipids.
Glycogen does store energy or no? No.
It does
What is?
store energy, but
Yeah.
it doesn't. What is it? Yeah, but it stores carbohydrate energy, not fat energy.
Okay.
Yeah. Fats in general, they have like their purpose. They have the most potential to store energy. So like, but if you're comparing between fats, carbs, and proteins in general, like if you see kind of like on the nutrition labels that you have, like you have like the calories per gram of like carbs or fats or protein. The fats, they always have the most calories associated with them. And so that's how you know they store the most energy. Does that make sense?
Yes.
Okay, sounds good. And now we have our passage. Did you have time to do a passage with us today?
Yes.
Awesome. So let me send that link over to you. Okay, so it's a PDF of a Khan Academy passage. So I just wanted you to be able to look at to go over the questions without seeing the answers, and then we can go over the answers once you're done. And it's 5 questions, and the passage is relatively short. How much time do you think you need to do it?
I would say like 10 minutes, or I mean maybe until like 9:10 at least.
Okay.
Because 9.
Okay. Let's do you want to do 9 minutes?
So like 9:12.
Yeah, 9:12. Yeah, so let's do 9:12 and best of luck. Hi, Rashina. Did you still need more time?
No, I think we can go over it now.
Okay. What did you think of the passage? Did you like it?
I think it was okay. It was pretty straightforward. It wasn't that difficult, I don't think.
Okay.
Mm-hmm.
Sounds good. Okay, so just gonna go over the answers really quickly. So question one, the answer was Roman numerals 1 and 2. So that means that it contained both alpha 1,4 linkages and alpha 1,6 linkages.
Mm-hmm.
So let me point out let me share this tab. I have the passage here. Let me just point out which ones are alpha 1,4 and which ones are alpha 1,6. So here, so you have 3 bonds. Do you see the 3 bonds?
Yes. Mm-hmm. Yeah.
Okay, so the one kind of the more top left corner one, that's so you have that's a 1,6 bond. So you have the left one, the left sugar is coming off on the carbon 1 side, and
Okay.
then the one on the right is coming off actually on carbon 6. So that's alpha 1. That's alpha 1,6, and then the other 2
Wait, can you
bonds.
just tell us really quick, how do we count the carbons? So is it from from the right one, the top one? How do you count the carbons, like the 6?
Okay, so
Do you start from
so
after
let's
the OH, like the branched off OH?
yeah, so the one with the branched off OH is carbon 6,
Oh, that's carbon
and
6.
then
Okay. Then
yeah,
it's
and then
5,
moving down
4,
5, 4, 3,
3,
2,
2,
1.
1.
Yeah.
Okay, so the
Mhm.
left of the ester is 1.
Yeah. So yeah, for me, I always say the anomeric carbon, so carbon 1 is to the right, usually the right of the oxygen within the the what is it? The cycle, not the cycle, the ring. There you go.
Okay.
Yeah. And then so moving on from there, so the one on
Wait, so then
the bond.
wait, I'm sorry. That is that the O with in between 2 carbons, is that a ester or ether? Like
I think
OROR.
that's an ether. Yeah,
Oh,
I
an
think an
ether. Okay.
ester is yeah, an ester is between, isn't it a carboxylic acid and an alcohol?
Maybe. Okay, so yeah, ROR is an ether, right? Okay.
Yeah.
So wouldn't the bond between the 2 glycogens on the right side, wouldn't that be 5 and 1?
On the right side,
Yeah, like the first one on the right. The first linkage.
no, no, that's actually carbon 4. So starting with the one in the middle, it's
Oh, it's
branching off to
4.
carbon
Okay, so this
1.
is a 4-1.
Yeah,
This is a 4-1 linkage.
yeah,
Okay. Okay.
yeah. So yeah, sorry on the other, yeah. So on this top one, you have the carbon 1 starting off on the left side. Yeah,
Okay, got it.
yeah. So it's probably easier to count backwards at which is 6,
Yeah.
and then yeah, yeah. And then do you see how the other bond is is alpha 1,4?
The bottom.
Mm-hmm.
Okay, this one's kind of difficult. Okay, so I see it here. So I guess this would be 6, 5, 4. Okay, 4, and then this is 6, 5, 4, 3, 2, 1. Okay, so this is 4, 1, 2.
Yeah, yeah.
Okay.
Yeah. So yeah, so you have a total of 2 1,4 bonds and 1,6 bond. So that's
Oh,
why,
okay. So it's the one
yeah.
6 on here on the
Mm-hmm.
left.
Mm-hmm.
5, 4, 3, 2, 1. 9. Oh yeah, 6. Okay, and this is the 1 6. Okay. Oh yeah, I see it.
Yeah.
Mm-hmm.
Yeah. Awesome. Awesome job. Okay, moving on to question 2. So the correct answer for this one was 2 molecules of glucose 1-phosphate would be produced. Oh wait, I keep forgetting to switch between tabs. So this is the answer to question 2. So the answer was 2. I'll just read off the answer here. So glycogen phosphorylase. So they're asking if glycogen phosphorylase came in and um, and cleaved bonds in the glycogen. So in the passage, so let me go back to the passage. So in the passage, it reads, glycogen phosphorylase catalyzes the cleavage of the glycosidic linkage between the anomeric carbon of one glucose subunit and the fourth position carbon of the adjacent glucose subunit. So what that means is that it cleaves 1,4 bonds. So what the question is asking is, how many 1,4 bonds would be cleaved, and as a result, how many glucose 1-phosphates would be produced? And the answer is 2. Does that make sense?
Is it 2 because we only have 2 1,4 bonds?
Exactly.
Okay. Yeah, because I put 4 because I thought we would have like 4 free glycogen units.
Yeah, um, yeah, that's why it's kind of important to study the molecule to see which is to see what kind of bonds are going on. Because yeah, glycogen phosphorylase, it breaks a certain bond. It doesn't break every bond.
Oh, okay. So it's the 1,4. So if we break 2 1,4 bonds, we only have we only have 2. And then
Mm-hmm.
2 units of glycogen will still be connected.
Yeah.
Okay. Mm.
Um,
Probably.
And then it didn't go over the other enzymes, but there's like other enzymes. So like glycogen synthase, it would actually form 1,4 bonds. So that's kind of like the opposite of glycogen phosphorylase. And then there's also 2 other enzymes that cover specifically that break and form 1,6 bonds. So there's like 2 enzymes for 1,4 bonds, and then there's 2 enzymes for 1,6 bonds. So it's
Okay.
not, it's not a universal system. Yeah, I just wanted to let you know for that. Let's see, question 3.
Mm-hmm.
Question 3, the answer is this one. So it's not true that large branch polymers of glucose are able to form chylomicrons more easily. The reason why, so I have the formal answer here, but the answer is like chylomicrons, they're actually made up of proteins and fats. So it's kind of irrelevant to glucose polymers or glycogen. And then going back to these other answers, the first one is true. Yeah. More branching allows for more hydrolysis to occur at the same time. And then the osmotic pressure one. It says osmotic pressure increases with molarity. So I guess the branching isn't relevant. That was like the one I was most shaky on. And then um, the one where they take up less volume and precipitate less easily is also true. So yeah, the last one is the only one that's pretty false.
Yeah, I think I got this wrong because I missed which of the following does not represent a possible explanation. So I put,
Ah,
I put C, large branch polymers of glucose take up less volume and precipitate less easily than large unbranched polymers. Because I thought it was asking which one is true and why would
mm-hmm.
that be the selective advantage, but it said which one does not. Okay, I just have to read
Yeah,
better.
I've definitely been there. Like even with like I remember taking like a ton of full lengths, and whenever that happened to me, I would just be like, ugh, like I did it again. So don't feel dumb for doing that. Answer to number 4 is GSD type 6. So they're asking which they described. So let's go back to the passage. So the symptoms of GSD type 1, which results from a deficiency in glucose 6 phosphatase. So a glucose 6 phosphatase is located in the liver, and it catalyzes conversion of glucose 6 phosphate to glucose. So that means it breaks off a phosphate. So going back to the table, what would that be the most similar to? So as I was saying before, phosphorylases, they break bonds. So phosphorylase checks off. So it'd be either type 5 or type 6. And then they specifically mentioned the liver. So the best answer would be GSD type 6. And yeah, that's pretty much all there is to that question. Did you have any concerns about it?
No, I actually got that one right.
Okay, awesome. Yeah, that's a pretty simple question. And then last question, the answer to the 5th question is 4 anomeric carbons. So basically, anomeric carbons are, let's share this one again. So the anomeric carbons, they're all of the carbon ones in the molecule. And since there's only four glucose molecules, that means there's only 4 anomeric carbons. Did you get that one right?
Yeah, I got that. Yeah, I got that one. At first I thought it was more complicated, but then I was like, you know what? It just can be. So yeah.
Yeah, I know. I think when I was doing it, I was like, am I overthinking this? And I was like, wait, no, this is actually really easy. So yeah, great job on the passage. I
Yeah.
hope
I was
you
like,
learned
is
something
this?
from
I'm
it.
like, I think it's 4, but I don't really know. And then I'm like, there's no calculations or math that I can do. I'm just gonna go with 4. So yeah.
That's a good guess too. Okay, so that's it for our passage. Did you have any questions about the passage? Is there anything in the text you wanted me to clarify?
No, I think everything was pretty straightforward. It's just, just the link. So I was a little bit late, but I'm glad I caught you. So yeah.
Okay, yeah, just make sure to work on your timing, and
Yeah.
yeah, you'll be good to go. Yeah.
Yeah, for sure. And I think because you still use, yeah. Oh, because it's still August, and I guess that new link is the September link. That's
Mm-hmm.
why, okay. She sent it with today's well, yeah, because today's 31st and tomorrow is the first. So tomorrow it'll be on the new link.
Exactly. Yeah.
Okay. Okay, there you go. Okay, perfect.
Yeah. I have a couple other slides for you before you
Okay.
go.
Mhm.
So here's some additional practice. I forgot to mention earlier. So the lipid stuff that we assigned wasn't necessarily relevant to what I taught. So here's the relevant videos to what I taught. The ones that were assigned were kind of more on metabolism, which you guys already went over in like class like 2 or something. So I just wanted to make sure that you knew about these videos. And then here's some extra sugar passages if you're up for it. They are more challenging. I gave you the easiest ones, so these are more challenging. And yeah.
Can you put those in the chat, please?
Yeah, of course.
Really
Let
quick,
me
yeah.
just let me stop sharing this. Okay, I think that's all the links. Do you see them?
Yes, I got them. I'm going to copy and paste. Okay, so these are some passages. Okay, perfect. All right, I'm just going to paste. Okay. Okay, thank you so much. This was the last biochem class of the course, right?
Yeah. Yeah, so this is the last biochem class. Yeah, so this is my thank you slide. And then I'll just unshare again. Yeah, it's the last biochem class. And then we have other classes going on for the month of September. We're able to get the schedule for that.
I think she said it'll be available tomorrow.
Okay, so I know there's like physics in there, and then there's the rest of physics, and there's the rest of second soc, and I believe the rest of biology. And I think that's everything in there. If I missed a class, I'm sorry.
Yeah, it's okay. And where
But
can we access like the recordings? Because in the email she sent the recordings for September, but like for August and stuff.
We have
I
a
think.
Google Drive with all the recordings. Oh, the recordings are sorry. The slides are in Google Drive. The recordings are in our YouTube channel.
What can you put the link if you have a chance? Because I couldn't find it.
Okay. I'm gonna talk to Sim to figure out how to get you connected because the videos are unlisted. So let me see if I can.
Oh,
Yeah, I'll try messaging you with like the playlist link to
yeah.
see
Thank
if
you
that
so
gives you
much.
access to everything.
Mhm.
But yeah, I'll work on that.
Okay, thank you so much. Because I was like Googling, I mean, I YouTube pre-med peers, MCAT peers, and nothing is, yeah, a whole bunch of other, MCAT videos and stuff came up, but you guys didn't come up.
Yeah, our videos aren't public, unfortunately.
Okay. Okay. So I'll just wait for the link. Thank you so much, Karen. See you
Yeah, thanks
next
for coming,
time.
Rashina.
You're
Yeah.
welcome. Okay. Bye.